Imagine a world in which we could create modified organs that wouldn't be rejected in transplant surgeries, engineering microbes to produce a larger quantity of essential medicine, and richer soils with biosensors that could detect pathogens long before they can affect our crops, make sugars that can act as a petroleum substitute to power our machines and cars. This world is not far away. Synthetic biology gives us the tools to reimagine our surroundings. Synthetic biology is the point at which engineering and biology meet. The standard components of synthetic biology, which include genes, or DNA sequences that code for specific biological functions, can be designed and modified, and then inserted into an organism like E. coli. These bacteria are then able to transcribe the modified genes and translate them into protein, such as insulin for human use in medicine, or biological sensors that can detect contaminants and pathogens as an early warning system. The building and redesigning of biological circuits is much like electrical circuits, where depending on its construction and modification can yield different products.
Synthetic biology allows us to make use of novel tools of biotechnology and engineering to tackle a myriad of global healthcare, environmental, and agricultural challenges. More than just a scientific field, synthetic biology is a community. It's composed of researchers and scientists from many scientific fields who come together to propose and deliver novel solutions for some of the world's greatest problems.
This may sound like a very general term, but it's likely that you have already learned about synthetic biology without knowing it. One tool currently being used in synthetic biology research is Crispr, a technology that enables you to add, edit, and remove genes much like puzzle pieces. In moving these pieces around, scientists have been able to create the first cloned mammal, Dolly the sheep, a popular scientific breakthrough you may already have heard of. Genetic engineering is a field at the heart of synthetic biology. Gene editing technologies like CRISPR allow us to edit the genomes of host organisms, such as microbes, to modify existing genes or to introduce new genes with new functions. This is accomplished through editing genes in the plasmid of a host bacterium, which is a small circular DNA found in the cytoplasm. For example, we can manipulate the plasmid of a wild-type E. coli bacterium to include genes for methylthionines, which are proteins that bind to elements like arsenic and cobalt that are present in drinking water. These are usually harmful heavy metals. This effectively traps these harmful chemicals within the bacterium and makes safer drinking water. We can also modify its biofilm-making capabilities to make the bacteria stick to water filtering systems so they can access the drinking water they will modify.
So, what does the future of synthetic biology look like? As the cost of DNA sequencing has decreased dramatically and continues to drop, research in genomics continues to become more affordable, and ideas that were once considered ambitious are now closer to being realized as reality. Synbio, as synthetic biology is sometimes called, is bringing forward significant advances. It's becoming part of the solution to many existing problems in medicine, bioremediation, and food and energy production. Although Synbio is a promising field with many applications, we must also give some thought to ethical considerations. We may be able to genetically engineer viruses to be effective vectors for gene therapies, or to modify the genomes of plants and microbes to endow them with more functions. But how ethical is it to do that? What are your thoughts on synthetic biology? Leave your questions and thoughts in the comments.